Hello everyone, I am Sangeeta Dwivedi, Assistant Professor of Modern Institute of Pharmaceutical Sciences Indore. Today I am going to talk about morphology of fungus, which is a subunit of Unit 3 and pharmaceutical microbiology. So let's start. So fungus basically is a domain of kingdom uh, Protista, and Protista kingdom basically divided into three domain. First is Protista, second one is algae, and third one is mold. So basically we known fungus we known as a mold also. So both are same, and the three domains are different somewhere in some their characteristics. That's why they are categorized in three different domain. So if we are talking about the basic difference between protista and fungus, so protestant protozoans are basically unicellular, while if we are talking about fungi, so they are multicellular organism. So this is the basic difference between protozoans and fungus. And the one more important thing which should be known about fungus, it that they have chitin in their cell wall. The main component of their cell wall is chitin, which is not present in plant and some other protists, and it is only present in fungus cell wall basically, and it is also absent in the cell wall of bacteria. So the study of fungus is known as mycology also, which is a sub branch of botany, and genetic studies have shown that fungus are more closely related towards animals as compared to plants. ऑलमोस्ट अगर हम बात करें तो देर आर वेरियस वास्ट मेजॉरिटी ऑफ स्पीसीज फाउंड इन अंडर द कैटेगरी ऑफ फंगस. Almost 1.5 to 5 million species are there in the category of fungus, but only 5 to 7% of them are formally classified till now. So in the starting of 18th to 19th century, a scientist named Carl Linnaeus and Hedrick Pearson and Magnus Fries have classified fungus according to their morphology or their physiology. So basic morphological characteristics of fungus are they are non-motile, and obviously they are eukaryotic organism, while we are talking about bacteria, they are eukaryotic organism. And the fungus can be differentiated in very differentiated on the basis of septi and mycelium, and they have a very differentiated nuclei which are surrounded by a nuclear membrane, and they can reproduce either by the budding or by forming spores, and they are heterotrophic in nature, while जो protozoans हैं they are they could be autotroph or they could be heterotroph, while fungus are always heterotrophic in nature. And what does it mean, heterotrophic, autotroph? Autotroph means they can make their food by their own, right? And heterotrophic means they are unable to produce their food by their own. So fungus are grow as a thread like filamentous microscopic structure which are known as hyphae, and these hyphae collectively are known as mycelium, and they variate in their size from 2 to 10 micrometer in diameter, and लेंथ में काफ़ी ज़्यादा भी हो सकते हैं. It should be centimeter, they can be in centimeter also in their length. So hyphae can be septate or non-septate. There are two type of hyphae basically. One is septate and the other one is non-septate. The non-septate type of hyphae is also known as sinusitic hyphae. So if the septa is present between the hyphae, then it is known as septate hyphae. If septa or compartment is absent in hyphae, then it is known as sinusitic or non-septate hyphae. So you can understand by this diagram also. So here if you can see, these small structures are known as hyphae, right? these structure are also coming here. They form a network, a strong network here, right? ये जो आप जानते हैं, जैसे ये जो मशरूम्स हैं, तो ये जो स्ट्रक्चर आपको ये दिख रहा है, इसके अंदर अगर हम डिटेल में बात करें तो इसमें एक हाईली नेटवर्क स्ट्रक्चर है ये इस तरीके का, जिसको हम जो ये मिल के बना है हाइफी से, राइट? और कंबाइंडली इस स्ट्रक्चर को हम माइसीलियम कहते हैं. दिस इज कंबाइंडली, कलेक्टिवली दिस इज नोन एज माइसीलियम. सो दीज हाइफीज आर ऑल्सो प्रेजेंट हियर ऑल्सो अंडर द ग्राउंड. यहाँ पर भी इनको ये हाइफी स्ट्रक्चर है, और कलेक्टिवली अगर हम बात करें तो इसको हम माइसीलियम कहते हैं. ये जो आपका अम्ब्रेला शेप का या टॉप पर ये जो कैप लाइक स्ट्रक्चर है, दैट इज़ स्पोर प्रोड्यूसिंग स्ट्रक्चर्स. ये स्पोर प्रोड्यूसिंग स्ट्रक्चर है, राइट? सो ये इस टाइप से हमको दिखता है, कैप लाइक और अम्ब्रेला शेप. Here I am showing the basic difference between fungus and bacteria. This is important by the point of view by your exam. So इसको आप देख लेना. There is no need to explain deeply here because हम बैक्टीरिया ऑलरेडी देख चुके हैं. So let's come on to the classification of fungus. There is two type of classification of fungus is there. One is depending upon the cell morphology. According to the cell morphology, fungus can be divided into four classes. First is mold, other one is yeast, third is yeast-like fungi, and the last one is dimorphic fungus. And on the basis of their sexual spore formation process, fungus can be divided into Phycomycetes, Ascomycetes, Basidiomycetes, and fungi imperfecti, right? So let's start by one. So molds are simply which can say a filamentous fungi are known as molds. So filaments of fungus is also known as hyphae, अभी जो हमने लास्ट में पढ़ा है. और I have already told you that if the hyphae are septate, means if they are divided into various compartments by the help of septa, then this is known as septed hyphae. And if the septa is absent in the structure of hyphae, then this kind of hyphae is known as non-septed hyphae or cenocytic hyphae. Like in the diagram it's showing, this one diagram is showing septed hyphae. If you can see, these white color structures are present in the hyphae, which is divided hyphae in various small compartments. और यहाँ पर जो ये नॉन सेप्टेड हाइफी है, यहाँ पे कोई भी कंपार्टमेंट जो है प्रेजेंट नहीं है. सो दिस इज द एग्जांपल ऑफ नॉन सेप्टेड हाइफी. ये एक जो ये पिक्चर है, ये शो कर रहा है बेसिक इस पिक्चर में जो बेसिक मोड शो किया गया है कि कैसे जो हाइफी होते हैं वो कैसे अपने आप को अडॉप्ट कर लेते हैं बाय फॉर द ट्रैपिंग एंड किलिंग ऑफ प्रे, राइट? सो ये जो स्ट्रक्चर आपको दिखाई दे रहा है, दिस वन, ये एक निमेटोड है, राइट? ये प्रे है. किसके लिए प्रे है? फॉर दिस फंगस. फंगस इस पर अटैक कर रही है. और ये जो राउंड स्ट्रक्चर इसके चारों तरफ, वो आप इसको पिक्चर को देख के समझ सकते हैं. ये फंगल का हाइफी है और ये कंपार्टमेंट में डिवाइडेड है, मीन्स ये आपके सेप्टा प्रेजेंट है. ये जो स्ट्रक्चर यहाँ पर दिखाई दे रहे हैं, ये किसी भी सेल के अंदर जाके उसको अंदर से कैप्चर करते हैं. इसको बोलते हैं हॉस्टेरिया, मतलब अपने खाने को अंदर लेना, ट्रैप करना, राइट? The second one type is yeast type, and yeast fungus are round and oval and elongated in shape. And here यहाँ पे अगर आप ध्यान दें तो the most important thing is this yeast type of fungus are unicellular, right? अभी तक हम जो भी बात कर रहे थे तो we are saying like fungus are multicellular, but this yeast are unicellular fungus. So they reproduce by the asexual process, which is known as budding, in which the cell develop protuberance in their top portion and enlarge by themselves and eventually separate out from the parent cells. So this process of asexual process is known as budding. When we are culture them, they form smooth and creamy colonies. For an example, like Cryptococcus neoformans and Saccharomyces cerevisiae type of yeast form these type of creamy and smooth colonies, and they produce asexually by the process of budding. The third one type is yeast-like fungi, and the difference is like if we are talking about yeast, the difference between yeast and yeast-like fungus are in the budding process. The bud is remain attached to the parent or mother cell in case of yeast-like fungus, while in yeast the bud formed from parent cell which is डिटैच फ्रॉम द पेरेंट और मदर सेल। जबकि ईस्ट लाइक फंगस में जो आपकी डॉटर बर्ड है वो पेरेंट्स सेल के साथ ही अटैच रहेगी, वो उससे डिटैच नहीं होती। फॉर एन एग्जाम्पल, कैंडिडा एल्बिकन्स इज एग्जाम्पल ऑफ ईस्ट लाइक फंगई। द लास्ट एंड फोर्थ फॉर्म ऑफ फंगस इज डाइमॉर्फिक फंगस। मेनली इट इज़ अ पैथोजेनिक स्पीसीज एंड जनरली जो पैथोजेनिक फंगस होती है दे एग्जिबिट डायमॉर्फिक फॉर्म। द नेम इट सेल्फ एक्सप्लेनिंग दैट डायमॉर्फिक मीन्स दिस टाइप ऑफ फंगस कैन चेंज देयर सेल्व्स इन ईस्ट ऑल्सो एंड as well as in form of fungus also, right? So if the temperature is around 37 degrees centigrade, fungus can be grow like yeast, and if the temperature is around 25 degrees centigrade, then they can show growth like in mold like fashion. For an example, histoplasma capsulatum. These all are the example of dimorphic fungi. The left side is showing yeast like growth, and the other one is showing the left one is showing fungus like growth and the right one is showing yeast like growth. So this is all for today. Thank you so much.